I'm Sam. I'm an assistant professor at the University of Cambridge, but I'm also a visiting researcher at EBI since 2019. And yeah, so today I'll be talking about polygenic scores, and it probably makes sense to first jump into what are polygenic scores, for those who don't know. Okay, so polygenic scores are basically a way for you to calculate your genetic predisposition to any disease or trait that's heritable. And so how that works is basically, usually we do these studies called genome-wide association studies, where we measure all the variants in your genome, and then we look at, for each copy of a variant of allele, how much does that change your trait value or your predisposition for disease? So then we do that across the whole genome. And what a polygenic score is then we take the variants that are associated with disease, we sort of pull them out, and then we basically aggregate those into a joint prediction of your genetic predisposition. And that's basically just a linear combination of how much that change in your DNA affects the trait value, and then how many copies of that allele, that genetic variant you have. Then you do that, you sum it all up, and that's your polygenic score. And for this scientist, their polygenic score for this this trait would be like 1.49. Okay, but that's just like a random number, and what does it do? Well, actually, these polygenic scores, if you look in a population of people, stratify them into sort of levels of genetic risk. And so, in a population, you get this normal distribution where some people are at the low end, most people are at average risk, and some people at the high end. But for traits and diseases, actually, this has like a big impact on your lifetime risk of disease. So here you can see that the people in this red part of the distribution, their polygenic score is highest for coronary artery disease, and their risk actually accumulates much earlier than people who are at average or low risk, which means it provides an opportunity to, target prevention. And that's just a general case, like for lots of different age-related disease, like Alzheimer's, different cancers. Okay, so at EBI, the main work I do is to develop this catalog called the Polygenic Score Catalog, and it's an open database with all the polygenic scores that are out there that are open and available to use. And so, it has information about how the polygenic scores were developed, who they were developed in, how well they've been reported to work. And then we provide the scoring file, this list of variants that you need to then go out and calculate a polygenic score on a new sample. Okay, but that doesn't really go far enough, because actually the tools and the expertise you need to calculate polygenic scores is quite challenging if you're not an expert geneticist. And really the power of polygenic scores will be about how clinicians and sort of less technical researchers can interact with them. And so, you would have really needed to do a lot of scripting, a lot of mapping, a lot of work with schedulers and stuff like that. And still, it wouldn't give you a good result. It would give you this sort of scaleless measure. So that's why we developed PGS Calc, or the Polygenic Score Calculator, and we chose to use Nextflow to implement that, because we wanted to basically abstract away all these really technical details into a reproducible pipeline that's can also be customized with lots of different parameters, but that basically abstracts away all these technical details and gives you the best practice to calculate polygenic scores. So basically there's, it basically can take any sort of target genome. So the genotyping data usually comes from a genotyping imputation, but it can also come from sequencing data. And so the pipeline allows inputs VCF files or the Plink files. Then the pipeline can either take polygenic scores from the catalog or your own polygenic scores that you want to calculate, and basically does all this technical variant matching, scoring, and aggregation, and outputs a report and the polygenic scores for you to use in your work. The pipelines are written using the NF core template, and then we test it to make sure it works with Docker, Singularity, and Conda in Nextflow. So many people just always ask us why not use Plink. That's the thing that people always use, but actually that still is really challenging for most users. And so we basically, and it's not scalable. You still have to script it all together, and that's basically what the pipeline does. It works with as many polygenic scores. It can calculate thousands of polygenic scores at once, and it works sort of the same for one sample as it does 500,000 people. It has sort of a simple command line interface to use. Basically, you input the data about where to find the genomes, what build they're in, and then which polygenic scores to calculate. It does everything and then outputs a report that basically has information about how the score matches your, how the variance in the scores match your genome. And it's all in a log. But actually, the big problem with polygenic scores is that they're sort of confounded by genetic ancestry. So this is just a graph showing you that, depending on sort of your ancestry, different ancestry groups like European ancestry populations or African ancestry populations will have these stratified polygenic score distributions that are really not informative. And so polygenic scores are on this like scaleless axis that's quite hard to challenge. So what we need is a method that actually goes into those distributions and reports your relative risk irrespective of your genetic ancestry. And there were no real tools to do this before. So that's why we built in sort of this version 2 of the Polygenic Score Calculator pipeline that you use with this run ancestry parameter. But basically all the data handling and variant matching are the same and the scoring remains the same, but we add new functions that using a reference panel of like sort of reference populations, you can basically identify the overlapping variants and do the PCA and scoring by matching. You do the PCA to make sure that you understand the ancestry of the people you want to calculate their polygenic scores on, and then apply these adjustments that actually remove the effect of genetic ancestry. And so now the new pipeline outputs a new report with information about genetic ancestry. So we project individuals, and you can see the sort of open circles are the query samples, the target genomes that you've added, and the dark circles here are the reference populations, which is a merger of 1,000 genomes and HGDP. And then you get information about the score distributions, these polygenic scores, and basically what we've implemented is 2 methods to basically make the distributions across ancestries overlap and not be so stratified, and then also report polygenic scores on this interpretable scale, basically your relative risk of a disease based on your genetics and based on people like you. And so I'm right on time. But yeah, this is the work of a larger team based at Cambridge, EBI, Baker in Australia, and funded by HDR UK, as well as NIH and multiple other funders. But thank you for your attention.
All right, thank you very much. Any questions?
How do you estimate which is the ancestry of each sample?
So we do it 2 ways. Basically, we use this like reference panel of populations. So like 1,000 genomes and HGDP, you merge them together, and those have sort of established population labels, like based on sort of the way they were sampled was to sort of have those ancestry labels. Then what we do is basically just you derive a PCA on those people, and then project the new samples into that PCA space.
How do you choose which SNPs to take?
That's just using the standard. You like prune to only common, prune to a small set of common variants that have a high math. So like, and then you can either use random forest or distance-based metrics to assign population labels. But actually, these are quite, in polygenic score analysis, we're trying to move away from that. These are actually very problematic to assign labels to people. And so actually the better methods now, which are implemented in the pipeline, use a continuous mapping of ancestry to decorrelate the polygenic scores from ancestry and put people on an interpretable scale. So we actually don't need to assign ancestry anymore to reduce the effects that you see of this ancestry stratification.
And how many principal components do you take to calculate this distance?
So in the ancestry decorrelation, usually people use 4 or 5, but you can customize that within the pipeline. It's not really a settled science.
Thank you very much.
No worries.